I want to thank professors and colleagues, professors that have come visited, professors that have taken the course, but especially colleagues and professors who send their students to Lakeside to take our Diatoms course. Of course the students, and we can't do this without a TA every year. And then the Friends of Lakeside, who have been really instrumental in supporting the classes, supporting tuition for the students, supporting scholarships and room and board stuff for them. And it's unbelievable. I want to start on a sad note. Since the last time I was here, we've actually lost three people that were really heavily involved in diatoms. Sarah Kingston passed away. Lane is our Kingston teaching fellow this year. And John Kingston passed away almost about 20 years ago. And Sarah, his widow, passed away just this last year. And they've been very supportive of Lakeside. Norman Andreessen passed away in 2019. He used to come out with Gene Stormer. He'd drive the van full of all of Stormer's gear out and hang out at Lakeside. So he was a presence here. And then sadly, Phil Woods was a student here in 2012, and he passed away last fall, tragically. So this is Lakeside Lab. I work at a place that's called the St. Croix Watershed Research Station. But our mothership is the Science Museum of Minnesota in downtown St. Paul, a big public exhibit museum, dinosaur bones, all that kind of stuff. We're located about 30 miles away from it, out in the middle of the country, at a small field station, much smaller than this. We have about this much land base, but much smaller in terms of buildings and stuff. We don't have an education program, so we don't bring students in. We have visiting graduate students and things like that, but it's a little different. It's still part of the field station, what's called the OBFS, the Organization of Biological Field Stations. But that's where I hang my hat every day when I go to work. Now, this is what I start teaching with Diatom class every year. I'm always worked up when I come down here to start teaching, and I can't sleep, and I wake up and I see the sunrise, everything. So the first thing I get to see is you guys didn't wake up from the sunrise. I always do. We have had, this is the 59th year that we've had what we always colloquially call it Diatom Camp. It was taught 63 to 2019. COVID kind of bit us in the butt. And last year in 2021, we had a virtual class. We didn't do an in-person class. And this year we're back. We've got 7 students, 2 visiting researchers, a TA. So we've got a nice full house up in the McBride lab there. So we have to make some definitions here. So some of you have probably never heard of diatoms. So let's talk about what diatoms are first, okay? So diatoms are one group of microscopic algae. They're very special. You can find them just about anywhere. Anywhere you have water or even moist soil, there will be diatoms living there. What's really special about diatoms is they have the ability to make a glass cell wall. So all the cell walls we're familiar with are based out of carbon. So this is cellulose up here. Diatoms make it out of glass. They make a 2-part glass cell wall that's put together just like a petri dish. They're not all round, of course. And you think you may not have seen them, but you actually have. So if you go down to Westlake Okoboji, anywhere along the shore, that's what it looks like, right? It's got rocks and they're kind of a brown color. That's diatoms. Diatoms are photosynthetic. They don't have, I shouldn't say they don't. They aren't green. They have chlorophyll, but the chlorophyll color is masked by other pigments that give them this golden brown color. So this is what they look like out in the real world, and everyone's seen that. You get yourself on a microscope, they look pretty under a microscope too. They look like that. You can see that golden brown color. To actually study them, we actually cook them or clean them, because what we really want to see is this glass cell wall that they have. And we put it under a microscope, and it looks just like this. They have different shapes and sizes and symmetry, and they're really important in the world. One of the things I like to say is that we look outside and we see all that chlorophyll doing photosynthesis. Well, the plants are doing half the photosynthesis. So that means every other breath you take is oxygen produced by plants. The other half are produced by algae, and diatoms produce about 20%. They produce 2/5 of that. So they produce about 20% of the algae, 20% of the oxygen. So that means every 5th breath you take, you darn well better thank a diatom. If you don't, I mean, then you have to stop breathing every 5th breath. They're the base of the food web. They're found everywhere. They're taking energy from the sun, doing a little photosynthesis, making some storage products, making a little fat. What does fish taste like? You say fish tastes like fish, right? Fish tastes like diatoms. Honest to God. Diatoms taste like fish, and little things eat the diatoms. The little bigger things eat eat those things. Minnows eat that. And minnows get eaten by a bigger fish. Fish actually taste like diatoms. So you can't hate that. What else can we do with diatoms? Who likes beer? Who likes wine? All right. So you go to any sort of small winery, small brewery, they filter their beer and wine to remove the yeast through dead diatoms. See all those little holes they have? They're perfect natural filters, and they use them to actually remove the things. Some other people use them for kill garden slugs and stuff like that. Because why would a slug eat a broken Coke bottle? The same thing would happen to you if you ate one. So that's why they use it as a thing. They're identifiable. Among a lot of the microscopic things, we can't identify them. But because diatoms have this really nice cell wall, you can identify them under the microscope. They have characteristics. That's what we learned in class. Since they're microscopic, they're also responsive. So when you change an environment, when you pollute a lake, the diatom communities there change. And we can monitor and measure that. That fossil, or that glass wall they have, preserves really well. So we can see fossils of diatoms. Tomorrow, our class is driving out to, which way is west? That way. We're driving out to Royal, Nebraska, and we're going to see the ashfall fossil beds out there. So they're 11.6 million years old, and we'll check out the rhinoceros. Yeah, that's pretty cool, whatever. We're actually interested in the diatoms that were living there at that time. Finally, they're tools. We use them in monitoring, we use them in paleolinology to study the history of things. They're great tools for environmental sciences. Now, so given that, this is the mission statement of my museum. I'm supposed to take something that's microscopic, something you can't see. I mean, I can't go like that. Here's I dropped it. And I'm supposed to inspire learning, I'm supposed to inform policy, and I'm supposed to improve lives with diatoms. We can do it. The other thing I wanted to define was a legacy. This is what Webster said it stood for, a gift received from a predecessor. And that's the other thing I want to talk about, is how what's happened in the past in this partnership between diatoms and Lakeside has left a legacy. One of the legacies that I have is this character right here, Dave Zarnechiae. He's taught at Lakeside in the past. He's passed away, unfortunately. When I was an undergraduate, here I am, I was 14 years old, and I took Dave Zarnechiae's freshwater algae course up at Lake Itasca's biostation. So he said, you know what? We should go to Lakeside. Charlie Reimer, famous diatomist, is teaching there, so he drove us down in a car, and we stayed for a night and met Charlie Reimer. And that was the first time I set foot on this campus. And this also was an inspirational time in my life. Field stations can do that. I was a lost cause at that age. I was a biochemistry major and didn't know what the heck I was going to ever do. And I went up to Itasca, and I left Itasca, and I was still a biochemistry major, but I finished that up, and I went into grad school, and I haven't looked back at biochemistry yet. All right, let's talk about Lakeside diatoms. The class began in 1963. A guy named John Dodd was a botany professor here on station down at Iowa State as well, and he taught, in addition to botany, he taught an algology course here for many, many years on campus up in McBride. Stormer, who was my PhD advisor, had just finished up his PhD in 1963, and Dodd said, Stormer, you should teach a diatom class. Stormer said, no one had ever taught a diatom class anywhere. And Stormer said, okay, but it's gonna be different. We're gonna call it a diatom clinic. All we're gonna do is we're gonna bring people who are interested in diatoms together to hang out, we'll learn together, we'll work on each other's projects and problems. And we did that. Stormer taught the class for 3 years, 63, 64, 65. And then he got a job at the University of Michigan, and they had to find someone to teach the course. So he reached out to Charlie Reimer, who was a curator at the Philadelphia Academy, and Charlie took on the course. And Charlie taught it for almost the next 25 years. And Charlie took the course and made it a little more, I'll just say a little more academic. It had tests and course requirements and stuff like that. It wasn't just a gathering of people. This 59 years, it started with Stormer, then came Charlie, and then Charlie gave it back to Stormer, so Stormer taught it in the 90s again. And then when we hit 2000, Stormer retired, handed off to Sarah. Sarah and I started teaching it in 2003 together. Sarah taught until 2014. Marina Potapova, who has Charlie's job at the Academy now, she taught it for a couple years. And then Sylvia Lee has been teaching it with me since 2015. So 60 years, 6 people have been the focus of this great partnership. And we truly stand on the shoulders of giants. Every one of those people is an incredible scientist. So what do we do in this diatom class? We get together, a group of students every year. We get together researchers who come and visit. We get together visiting people. We got Joe Mohan, who's come visiting up at, he's taking us out to Ashfall tomorrow. And we learn about the same things that I think Charlie probably taught about. We learn how to use microscopes, how they work, what we can do with them, how we're going to look at diatoms. We learn about collecting. We learn about how to collect diatoms. We learn about diversity of diatoms and their systematics, how these trees of life are put together. We talk about the history of their study, the nomenclature, how we put names on diatoms. We talk about ecology, what they mean. We talk about how they're used as research tools. We bring people in to talk about that. And the most important thing we do is we build a community. We build a community here on campus of nerds who study diatoms. I mean, and recognize that this is the place to do it. That's my class from this year. Oh, and one thing I want to point out, this is normal. This is typical Friday night, 10 o'clock at night, and the lights are on at McBride. Not because people need to study diatoms, because they enjoy studying diatoms. All right, let me talk a little bit about this partnership between diatoms and lakesites and what this legacy is. I'm going to talk about 3 legacies. We're going to talk about how we use Lakeside to spread the news and train the world in diatoms. We're going to talk about how we've used this partnership to discover the biodiversity of the earth. And we're going to talk about how this partnership is really leading the forefront of science. So let's start with training the world. Every year we have the class, we get students and visitors from all over. I looked through the list, we've had students from 17 different countries, visitors from more of them. We get students who aren't just grad students. We have undergrads to professionals. I've had people who are retired come and take the class because they wanted to learn diatoms. Postdocs, professors even, who come and want to take diatoms. And again, what we're trying to do is build a community of people who are supporting one another in the study of diatoms. Here's what it really means. In 1963, when Stormer was asked to teach that course, I sat down and I beat my head to try to figure out how many people were studying diatoms in the US. The answer was 6. There was 2 at Philadelphia. There was 1 at the California Academy of Science. There was 2 at the USGS that studied fossil diatoms, and there was 1 amateur person up in Washington State. That's it. 1963, and Stormer. We have changed that. We have used this partnership to develop something bigger than that. And it shows. We got something really special this year. We have a 5th academic generation student in the class. So we learned about Dodd, right? Then Stormer taught the course. So that was Dodd, Dodd's student Stormer. Jan Stevenson came and took the course. He was a student of Stormer's. Stevenson had a student, Kevin Wyatt. He took the course in 2005. He's now at Ball State University. And this year, we have Cassie I blew it. Just like that. Arroyo. Damn it. What is it? This is more on Facebook. Cassie is joining us this year. Is a 5th generation Lakesider. I don't know how many classes can claim one of those. We've got, I think we got a 3rd generation here as well this year. What this has done is populated the world. So in 1963 we had 6. And did you listen close? How many were at a university in 1963? 0. This was my quick count and a picture of their thing of every one of these represents someone who came through Lakeside, took the Diatom class, and is now a professor at one of these universities. And they're not just, they're using diatoms. They're training their students. Their grad students and undergrads are learning about diatoms. That's the impact that this place has had. It has changed academia to include diatoms. What's our second legacy? Biodiversity. We learn about it in the class. We look at diatoms, we try to understand, what, we can look at them alive, we can look at them dead. And what we're really trying to do is train students to see diatoms. And then what we're really trying to do is this. We're trying to get them to design a t-shirt, but we're really trying to get them to think like a diatom. So, and as part of this process, we see new stuff that no one has ever seen before. And it's a rare opportunity. We get students who are bringing material from places that no one's ever gone before and bringing it to Lakeside and finally having a community to look at this stuff. And we find incredible new things in there. There have been 35 brand new species that have been described and discovered by the students working at Lakeside as part of in the 60 years. They're not all from Iowa. There's 17 of them that have been described right from Iowa here. But so we're discovering this biodiversity. In our class, it shows. Here's one that was described in 2010 by one of the master's students and an undergrad in the class. Here's one, we had a student from Iran who came, brought material, threw it under the microscope. It was like, what the heck? She described a brand new species. Ian Bishop described this one from out in Oregon. Another student who was from Australia brought this material and had us look at it. You can probably guess where this guy was from. Maryland, named it after his collecting site. It goes on. Here's one from Arkansas that was described. We had an undergraduate a couple years ago. Jackie was her name. She sat down. Someone had told her to take the diatom class. Like, why did? Well, they just said it was good. I should take it. She had no. She sat through the first day of class, and she gets done, and she looks at me and she says, "I don't get it. What's a diatom?" They're like, "Okay." And by the end of the class, we had found a little problem for her to work on, and she had published a paper on it. And she's the first author. And finally, I'm going to come talk a little bit more about this, but this is a website that is a direct product of Lakeside Lab. It's called Diatoms.org. DONA stands for Diatoms of North America. And it is, I'll talk about it a little more, but we require the students to do it as their final project. And every one of those students gets a publication. It's an electronic publication, citable, peer-reviewed, ends up on a website, and it's done by the students. And we've had about 100 contributions to this website from the students. The students are still doing it. last year we had a virtual class. It's like, come on, you're not even required to look through a microscope. How are we going to find anything cool to look on? Well, Sylvia taught the course. I gave a lecture on life histories, and I showed lots of cool stuff. Heidi, who's a student at University of Montana, she had taken the class virtually, and after the class gets done, I get this email. And she said, I'm growing cultures. I got these cultures growing, and she's working on this thing called epithemia. And what's cool about epithemia is internally they have another group of algae that live inside of them that helps them get nitrogen. So it's a cyanobacteria. And she said, all my cultures look terrible. Look at these things. They got funny. They look gross. They're looking nasty on me. What's going on with this thing? And I said, oh, you've got internal valves in your thing. I've never seen that before. And so we searched the literature, did the work, and 2 weeks ago she submitted a manuscript on it. It's not even part of her dissertation. Another one, this is David Burge. He was a student in the class in 2012. He was the TA for us in 2013. He came back in 2017 and was what's called the EFS, the E.F. Stormer Fellow. So he got some money to come do research here. We had collected a diatom up on the Apostle Islands, and David wanted to work on it. What he ended up doing was something called shape analysis. So we looked at all these populations of diatoms, and he wanted to, there's 4 different populations, and he wanted to try to separate them and see if we could do it. That's what he did. And in the end, the evidence said, this is something different. You know, he said, this is one species here, this is a species, and here's our species. And we used other evidence as well, but he ended up describing it. We call it Semiorbis eliaceae. It's an extremely rare genus, never been reported in Iowa. I can say that with confidence. Finally, I would like you to meet Iowa's newest diatom. This is great. So this is a diatom that was described last year. It's called Coconias Zarnechiae. You've heard that name before, and I showed this picture. This is Dave Zarnecki, who taught here. Well, one of the things Dave did, he was passionate about culturing algae. He wanted to, he didn't want to just study them under a microscope. He wanted to culture them and see them alive and grow them. He passed away in 2006, and a bunch of his students, all of us Lakesiders, by the way, drove to Dubuque. He taught at Loras College. We packed up his culture collection and we shipped it down to the University of Texas. Well, in 1982, he was here on campus and gone down to the dock here, and he had cultured this diatom here. It's a Coconias. And it's been sitting, growing in the culture collection down in Texas. And a group in Germany and one in California got together, and they requested this culture. They did molecular analysis of it. They looked at its morphology. They looked at it alive. They even looked at it having sex. And in the end, all the evidence proved that this is something different than any other Coconias, and they described it in honor of Dave Zarnecki. So that's it. That's the newest diatom that's been described from West Lake Okoboji. So that's kind of cool. Last, we have a bigger picture. We have had students here, and then we've cast our net around the world, right? Well, this is a Prussian named Ehrenberg, and he was working in the mid-1800s, kind of in that Victorian era when there was explorations going out. His task in life was to describe every microscopic organism in the world. And if you look at his stuff, you can see that. Here's a picture, this is under his microscope. Well, this is from Niagara Falls. This is from the Rhine River in Europe. This is from the Ganges. So he was getting samples from all over, sorting out these things and describing it. In the end, he described 2,055 species of diatoms. Now, 400 students have come through Iowa, or Lakeside diatom class. How many species do you think those students have discovered and described? That's your comparison, 2,055. Anyone got a guess?
500.
500. Anyone? 200. 1,300 species have been described by the folks that have been discovered and described, documenting the biodiversity on our Earth. And we know that we're in a crisis, losing things as fast as we can. Well, if we're losing them, we better figure out what they are before they go. So that's been a big component. All right, final thing, the final Lakeside legacy, what this partnership has done, is that we are, the class and this partnership has developed into something really big. I talked about this DONA project, Diatoms.org. Here's the website when you open it up, and here's what it's called. It's a collaborative work in progress, growing and changing as the science advances. Just in January, we finally published sort of the first time we'd actually published a paper describing it and laying out what our dreams for this site was. What it is it's a tool. We go up in our lab there, and there's 400 books in there, and most of them cost between $200 and $300. So what we've done is translated all this information to a website that anyone can use. It began because of this. Every year we bring up a course pack, and Sarah, and Sylvia, and I, and Marina, we're trying to keep this up to date with all of the things that are changing in the world. So every year we bring this thing and present it to the students, and finally it was like, you know what, we can make those kind of updates on the web. Let's develop this resource on the web. And it started out with taking how we treat a genus, so everything's got a genus species name, and we took the information from our course pack, created a website, and allowed us then to look at the differences among the genera. Then we tackled the species. So here's the next thing. We needed content to look at how we could differentiate species in there. Here's a bunch of different species within one genus. And the contributors of this have been students, of course. It's professors, it's researchers, it's retirees who are just passionate about this stuff. It's amateurs. There's still amateurs out there. And we've even used citizen scientists to do this type of work. Importantly, it's always involved students. I mentioned that this has been, it's been since I think 2010 or 11, I think 11. We've made it the final project for the students. They are to create a species page of their own and prove to the world that they are the experts, submit it for peer review, and get it published. The students all over the years have been coming in and doing this. We ask them to take their collections, look at them carefully, add some information about what it looked like alive, maybe where they collected it. There's a lazy lagoon over there. We've all been there. And create that species page themselves. We ask them to, take pictures of it, write a description, find out about the ecology using the literature of this stuff, and find the original description of it. In the end, it goes public. There's a citation that they can add to their resume, and they're a published author by the time, within a couple weeks after finishing the class. No. What this has done is really changed how things are in our world. I mentioned we have 400 books up there. Some of them are truly priceless, unavailable. You have to buy them through antique dealers. They're important still, but what we've done is created something more. When we evaluate science, these are the words that we teach our students, that we want science to be transparent. We want to know what you did. We want it to be reproducible. We want to be able to take your stuff and do it ourselves and come to the same conclusion. We want it to be defensible. Know what you did and be able to stand by it. But what we don't ever think about is this, accessibility. Do we have access to our science? And I would say in general, no. You have to join a society. You have to have access to a university library to access science. And we've changed that through one website that came out of Lakeside Lab. It's sick. 2 years of page views. We're getting 1 million hits a year on this website of people looking up diatom stuff, learning about it. And it's not just, it's in the US, but it's all over the world where people are accessing this site. A way that, in our paper we wrote, when knowledge and data are more accessible to everyone, science becomes more equitable. And that's a really important thing that we need to strive for these days. Last, it's a responsive thing. One of the things that happened in the last 6, 5, 7 years is we've had big federal programs using diatoms to monitor, and we've run into problems with it. It turns out that each of the people who analyze diatoms comes up with a kind of a different answer. And it's called Analyst Bias. And it depends on how good your microscope is, who your advisor was, what kind of literature you have in front of you. And so one of the things that DONA does is it provides a flora, a taxonomy that everyone can get onto, and they don't have to buy a $400 book. The other thing that we've done is sort of changed how we've approached diatoms. Rather than look at a diatom and try to identify it, we say, let's group them together, kind of give them a pet name for right now. We'll figure out exactly who they are later. But it takes a lot of this bias out of the process by giving you an easier tool to do it. So we've developed that. Last, COVID, of course, threw everyone a curveball. And one of the things that we did is we said, man, this is crazy. We're all stuck at home. We don't know what to do. We said, we're gonna institute something that helped keep this community together during COVID. So we instituted something called the Diatom Web Academy. And what we do is every 2 weeks we do a seminar on diatoms. And some of them are, very technical, how to identify this group, whatever, something like that. But others are fun. We have one called the Art of Diatoms by an artist down in Florida who's used diatoms in his art. Joe over here did one on three-dimensional diatom reproduction. That's his image there in the back. And that's what we've been doing for 59 years. Next year's the 60th year, Mary. And I don't know, but you should turn this place upside down with diatoms. How does it happen? How does it happen? Number one, it is passion. It is a bunch of us that are we love this stuff. We love diatoms. And it's the students, it's the professors, it's a community that's supporting us that is going all over the world and making a difference. We do it by support. We've got 3 scholarships that are managed by the Friends Group. They were created by the families of past diatomists who set them up. But we use those to support students through an especially important one is the Kingston Teaching Assistant Fellowship. Lane is this year. Where are you at, Lane? You're right there. Lane's our teaching assistant this year. Dude, you need a haircut. We get a good deal of support from the Friends Group, especially with room and board scholarships. I think almost everyone in the class has a room and board covered this year. And of course, Lakeside Lab is instrumental in this. It's great. We send them a shopping list. We send them like, oh my God, here we come again. And it's ready for us to go every year. And in the end, this partnership means that we're doing better science, which is really, really, really important. So, thank you.
I didn't see the Twitter one. Do we have questions for Mark? Out there.
Yeah.
Someplace I got to, maybe one of these previous lessons, that you could do deep coring and get diatoms, which would allow you to, a long time ago, recreate climate or weather stuff.
Yeah, one of the things that we do a lot of is recreate histories of lakes. One of our students, Adam Heathcote, he was in the 2000s, he cored all of the natural lakes in Iowa and looked at their history over time, including West Lake. Stormer did West Lake Okoboji, but that's oftentimes what we're doing to try to understand what they were like maybe before Europeans arrived, and how they've changed in that time. But we can go longer. I mean, every lake in this area, got overrun by the glaciers and was formed by the glaciers. There's a few oxbows, that's different. But each of those has a 13,000-year history of sediment buried that we can sample. We've got a student this year, Sav, is working on, is it a volcano crater? Yeah, so it's an extinct volcano crater that used to be a lake in New Mexico. They've dug they've dug a core out of it. It's all dried up now, but the old mud is still there. And she's working on diatoms that are 400,000 years old, trying to understand what the environment was like back then. So that's one of our major uses of diatoms. Thanks. Yeah.
What's the criteria for sharing knowledge and putting it on the website? Someone review that, or?
Yeah, so the website is managed, is that probably the right word? It's housed at the University of Colorado, and it's managed by we have an editorial review board of I think there's 7 or 8 of us. They were the authors on that paper. And interestingly enough, the authors on that paper, there is not a one of them who hasn't been to Lakeside. Either taught here, a student here, or one of them has been a visitor here a few times. She hasn't actually taught here, been a student. But the information is put in. If someone adds content to it, you set you up as a user for it, or contributed to it. And if you contribute stuff, it's reviewed by other colleagues just to double check that it's good, change it a little bit if it needs to be, and then it goes public.
Other questions? Can you say what some of the uses of other than that they help us breathe, what are the uses of diatoms?
We used to always talk about how they were part of toothpaste. They don't really use it in toothpaste anymore. They got that little gritty feeling you get. Used to be diatoms. I don't even think I've been around when they had diatoms in toothpaste. But other uses for them are really for environmental monitoring. They're a great tool for there's big stream monitoring programs and lake monitoring programs. I use them for monitoring. We're part of a program in it's called the Great Lakes Network Parks. So we work on the national parks that are around our Great Lakes: Indiana Dunes, Sleeping Bear, Pictured Rocks, Isle Royale, Voyageurs, Mississippi St. Croix Waterway, using the diatoms to look at water quality changes that are occurring in those systems over time. Paleolinology, the study of sediments, is one of the main uses of them. It's a field that has also exploded as we want to understand how the changes we're seeing in the world now fit in the context of what changes have occurred in the past and what we might be expecting in the future as well. There are industrial uses for them. Diatoms have been around for about 200 million years. And there are huge deposits, fossil deposits out in California and Nevada that are actually actively mined. That's where you get this stuff called diatomaceous earth from. And it's used for additives for concrete, for fireproofing. It's used as an abrasive. If you ever had your mother ask you to polish the silver, you were using diatoms to do it. It's used as additives for reflective content to paint. And it's used for things like filtering media. I told the students a few days ago that I actually met my first diatom at McDonald's. And it's true. So I was in a class in college, and the teacher gave a lecture, and it was the first time I'd ever been introduced to diatoms, and they did the same spiel. They said, you use them for filtering. And I was working at a McDonald's at the time, and every day we would take this machine, hook up our fry vat to it, and we'd put this piece of paper and we'd sprinkle this white powder in there. Ding! Went to work that day, did white powder, didn't get busted, put it under the microscope, fossil diatoms. So, met my first diatom at McDonald's. Yeah?
So you talked about the 6 people that have kind of been the instructors of this knowledge here at Lakeside, but it's obviously spread through the interaction with students and the community. Can you talk what about this particular space has made it such a petri dish, if you will, for that kind of knowledge spread and that kind of home base to be built?
Yeah, I mean, I think it started out as just don't want to say a one-off, but a unique place where people could come to learn diatoms where they weren't learning them anywhere else. When we look at those early class lists, it was people working for the Hawk testing company that were coming and taking the class. There was people who were agency people, working for the USGS or EPA who were coming, because they wanted to start learning about diatoms and how to do it. And then of course there were students who wanted to apply it in their own stuff. It's become, I think the reason it works is it's a unique resource. If you head up to McBride, you'll stick your head in the door and there's 12 microscopes set up on the tables in there. And the first with a digital camera sitting on top, hooked up to it, so that the students are coming in and they're exposed to exactly what they need, exactly what they're going to need to get their work done and learn about diatoms and use them in their work. Many of them, they're going to go back to their home institution or their home base, and they're going to have a poorer microscope than they have right here, and it's the truth. And we expose them to a really high-end microscope that's knock your socks off cool. We bring in literature, so we have this history of people working on diatoms. We bring in 400 books up there from Iowa State University library system. I can't imagine what they must think when we get that list every year. Oh, these guys again. Here we go. And they got to recall them from people that have them on campus and stuff, and we bring it up. And right now in McBride is the 4th best diatom library in the country. Best one's in Philadelphia. I might have a really good one. There's a really good one in Colorado, and then there's that one. So, but that's the type of thing. It is a unique resource that is made available.
And clearly it's the people. I mean, when you give a presentation and you see the passion,
Yeah.
I mean, the people are very special.
Yeah.
What's the future?
What's the
What do you
future?
see in 5 years?
We continue. Our website is really continuing to be the thing. It has become not just a tool for us to use, but it's become the envy of the world. There's other people that have tried to do this type of thing, to create resources that are really driving the science and helping the science, and they're looking at this as the tool to copy or emulate around the world. For us, it is probably, I would expect that we're going to have to figure out how to bring molecular biology into it at the station here as part of the training set. That's one thing we have not included in the stuff here.
So nodding heads over there.
Yeah, and so I think that is I'm not a molecular biologist myself. Would love to learn it, but I think that's going to be, my friend and colleague, and I hired him, David, who was the TA many years ago. He came down and talked last week to the group, and he's been using some molecular stuff. So I think we may start seeing it here, either working on data sets or doing some of that. But it's really, it starts at the basics of where's the power switch on that microscope, and how are we going to make sure we're using it right. Yeah.
Okay, so a couple slides back, I think it was like a text box under where you're talking about like the analyst bias. There's like some type of connection between taxonomy and phosphorus.
Oh,
I think it was saying.
This one.
Yeah.
Yeah.
So I don't know. I guess could you like explain a bit more what that's about and how like our manipulation of nutrient phosphorus can impact that?
Yeah. What Sylvia had to do is she had to go into this isn't the data. This is actually showing how single analysts, their data are grouped. That's what that's showing. But what Sylvia had to do is go into some of these data sets that have been analyzed by multiple people, and she had to make all kinds of choices about how to handle this taxonomy. Sometimes it was wrong names applied and she had to combine them. Sometimes it was just like, you know what, we're never going to figure out what all these people did. We're just going to call it Ignanthes, a genus level stuff. And what she did is she used that data to prove that it wasn't unusable data and could still help us understand phosphorus gradients in the environmental samples that were analyzed. But it also was this kicking off to figure out how we can do it better and how we can use our tool to do it better. Does that make sense?
Yeah.
Yeah, thanks.
Any other questions for Mark? If not, thank you so much, Mark. And again, we welcome you to come down to the dining hall. Mark's probably itching to get back to the microscope as